Good morning, everyone. I would like to introduce and invite the speaker of the Animal Experimental Unit, Dr. Ganesh Vythilingam. He is a pediatric surgeon and a senior lecturer at the Department of Surgery of our faculty. He holds a PhD in urological tissue engineering. Right now, Dr. Ganesh is undergoing his clinical fellowship at the Great Ormond Street Children's Hospital in London. So we are very fortunate that he came back shortly in KL for a conference, and he's able to give us his experience of running animal experimental procedures in AEU and involving a international team, right, from Switzerland. So Dr. Ganesh, over to you.
Thank you very much, Prof. Wong, and good morning, everybody. So let me start by sharing my screen. Okay, good morning, everybody. And my topic today was is Rapid to Man, Switzerland and Malaysia, the journey in clinical translation. So I'll share with you a little bit of my work in PhD and how I actually worked at trying to get clinical translation done, and especially with the help of the Animal Experimental Unit. The clinical problem that we were trying to solve at that time, that was part of my PhD work, was dealing with the urethra. As a pediatric surgeon, I have patients who have hypospadias, which is on the left-hand side of the screen. It's a congenital anomaly. It's seen in 1 in 250 live male birds. Adult urologists, my other colleagues, tend to see urethral stricture relatively quite prevalent in Malaysia due to motorcycles and when they'd have pelvic fractures. Previously used to be due to infections, but that's come down. But generally trauma seems to be the biggest problem. And urethral reconstruction is not easy. The current solution is sometimes you need to use buccal mucosa if you don't have enough tissue. Therefore, you need to harvest it from the oral cavity. Patients have 2 surgical sites. You've got a situation whereby they can develop paresthesia in the oral. And the thing is, with urethral work, you sometimes do have a risk of having a recurrence, and sometimes it can be up to a 20% complication, and then you need to harvest more tissue. So you've got very limited space in your oral cavity, if you can imagine. People were talking about tissue engineering. This is even way back in the early 90s, early 2000s. We're looking at tissue engineering as an option. And the problem lies in the fact that it's a large hurdle to get across. Things with acellular grafts were not really doing very well. The literature was showing the fact that defects longer than 1 centimeter were not actually getting regenerated, and that's really not giving us as a surgeon an alternative. You know, cell-based technology was there, but it's just very difficult to translate clinically. And that was my whole PhD question, and why in 2014, I actually went over to Switzerland to start my PhD in the lab of Professor Jeff Hubble. And in his lab, there was a lab under Professor Peter Frey that looked at experimental pediatric urology. So this is Lausanne. It's very close to Geneva, train ride from Geneva, beautiful place. And I basically spent my PhD working in that laboratory with a lot of engineers. And this is them, the 2 senior scientists who basically were my postdocs who taught me research. And together we looked at, my research topic was looking at how to engineer a graft for application in urethral surgery. And we wanted to make it simple, something without the incorporation of cells and bioactive molecules. And the hope was with such a method, we could at least translate it and make it more cost effective for clinical translation. This was the first graft that we fabricated. It was the DCGT, which what we named it was a double collagen tubule graft fabrication. We actually used a technology that was previously utilized in the lab and actually developed in other centers where we used liquid collagen. We neutralized it, and we used a molding technology to fabricate this graft. And literally, it sort of looked like a little bit very jelly-like, say the least. And taking from the previous talk, we looked at electron microscopy. Now, I'm a clinician, never did anything with looking at a microscope, and yet working with the team there, I mean, I got this imaging taken by my colleague who helped me with it, trying to understand the surface structure and mechanical properties, because it was not a very strong material, biomaterial we created. But we learned, when I looked at these images, there was a duality due to our fabrication method, if you can look at it. And again, fun things were also understanding the, the size of the material, the porosity. And then we did our rabbit model, and that's where I got the help from AEU. We came up with this idea of using the rabbit. Other animal models, obviously, the smaller models like the mouse and the rabbit, too small. And what we said was, a normal rabbit urethra, we will remove and replace it with our tubular collagen scaffold. And the hope was the fact that over time, we could have more cell infiltration, and the acellular collagen scaffold is remodeled by the host rabbit, and we should get a functional model. This is the work. This was circa around the time of 2015. I used to come back to do my animal experimental work in AEU. That's my senior scientist. We could see in the corner, left-hand side, Dr. Larson, who flew down with us, and we did our animal experimental work here. And it was done with two reasons why we actually used the AEU. For one thing was the fact that it's very important. Rabbits are very expensive animal models to work with, and you have to make sure they survive the initial period. They're very labile. And the fact was, in Switzerland, we were doing it ourselves in the university hospital there, but they were not surviving. We were in the immediate post-operative period, or even on table, these rabbits were not surviving, and they had better outcomes here. So we did all our animal experiments here in the AEU, and this is what we did. We basically resected a segment, and we used our collagen scaffold. If you look at image C there, literally looked like a very delicate piece of tube, and we anastomosed it. Very important in animal experimental work, you got to check on your animals. It's not doing it and depending on other people to take care of it. Though the AEU provides a very good environment, you still need to, and we had our own checklist. This is an example of that. We made sure we monitored them. I had to go in every day to look at this and at the animals and make sure that the animals were surviving in order for us to see what happens with our graft. This is what we found in our first graph, this surgical outcome. So we had a 60% no complication. However, we had 40% complication. Not great. The current gold standard, as I said, was buccal mucosa, and we were looking at 21% at the upper limit. So we've created a biomaterial that was not really doing, what it was supposed to do, in the sense that we were hoping that it didn't have any fistula. And a fistula is basically an abnormal opening, and a stricture, which is basically a narrowing. So we were not achieving what we were hoping to achieve with the biomaterial. But again, interesting to note from the previous lecture, same thing, I spent a lot of time looking at pathology slides to understand how the tissue was regenerating and correlating with the biomaterial. And we're looking at different time points and understanding how the smooth muscle cells were coming in and how the urothelial cells were coming in. And we sort of understood that time was an important factor for regeneration. And again, we looked at immunohistochemistry at different time points and realized, and if you look at the 1 month, 3 months, 6 months, and 9 months, you sort of get the uroplaking 2, which is the green color, somewhat coming relatively early on at 1 month and 3 months. But the red color, which is the smooth muscle actin for smooth muscle cells, were coming in much later at 6 months and 9 months. And you have to compare it with normal rabbit urethra. So we were still way off, but we utilized this information and we said, well, you could use a tubular acellular graft, and it can be sutured and regenerate a 2 centimeter defect. As I said before that, they said a tubular 1 centimeter defect cannot be regenerated. We did have 40% complications, but 60% were fine. And we noted the urothelial layer was the first layer to complete regeneration by 3 months, but smooth muscle cells were only coming in as early as much later. So it was a time-dependent regeneration. So back to the drawing board, again, using electron microscopy, again, changing the materials. I mean, I probably have skipped a couple of generations of graphs that we're working in the lab. again, we correlated with what we did with the electron microscopy to understand the porosity and looking at how we could improve the quality of the material and correlating it with what we understood with what we saw in immunohistochemistry and also our histology. And this was the 5th version where we came up with this low density graph and high density graph. And again, what we saw was where the preferential cells were coming in. And one of the things that we sort of figured out was the fact with the low density graph, we were sort of getting more smooth muscle cells coming in much earlier compared to the high density, which actually was attracting more of the urothelial cells. So again, we got this information, we actually put it in the implantation. We found out that the low density graph also had no complications and were having a better regenerative potential. So what we did and what we concluded at that time was the low density graph exhibited a better functional result, better ingrowth of smooth muscle cells, and we had a 90% no complication rate, and hence early smooth muscle regeneration is an advantage. And obviously at this stage, it was a lot of time just looking at pathology slides, looking again over and over again and how we could understand the regeneration potential. And again, this was primarily done with all the rabbit experiments. We had worked about 98 rabbits experiments, and as we did everything in AEU, it was important to keep these animals long enough. Some were even kept up to 1 year to understand the journey and how the regeneration takes place. So we've come to the state where we pretty much understood where we were, and we needed to come up with a large animal study if we really wanted to take it to the next clinical translation. Now, this was somewhere around 2017. I started my PhD at 2014. In 2017, we were looking at large animal models. And thankfully, through AEU, we got in touch with people in UPM, and we started our large animal study there. And this was us operating in UPM. We tried, the goat model didn't quite work. And then we actually ended up using the dog model, was a standard model for urethral work. As you can see, that's a picture of Professor Tambadure and Dato Selva doing the surgery, and myself and Dato Selva actually scoping the dogs in UPM. Now, at the same time, the collaborators in Switzerland were really happy with the work being done in and the AEU, that we started funding other experiments with regards to bladder regeneration. And that was done concurrently there. Now, these were the preliminary results of our work with the dog model in UPM. We did 3. It was a challenge. It's not easy working with animals. Again, I must stress that I got kicked by a goat and I got nearly bitten by the dogs. So it was a lot of, trying to manage them, but the removal of catheters, which the animals will be doing it. But we managed to get early data that showed that a 4-centimeter graft could regenerate. We did ultrasounds, and then again, we look at histological analysis. We went up with the models up to 1 year. Again, we found that the grafted area was regenerating. We did endoscopic examination, and concurrently, if you look at the right-hand side of the screen, we did bladder implantation studies. Using the bladder model, again, there was a need for patients who had a bladder reconstruction being done for a graft. So we were working on that in the AEU. Now, apart from just in the urology field from Switzerland, we had new collaboration coming in to University of Malaya. This was actually in cartilage work. So a lab in Lausanne in EPFL were looking at a place, wanted to reproduce good outcomes in large animal models and animal models, and we actually got them to come here to work with our team because they felt that they were getting better outcomes. So this was with the orthopedic team from Lausanne. By this time, it was 2018, we had large animal model data. I had actually presented this data to the ethical board in UMMC. Professor Louis was actually the chairperson of the committee, and I had to present all of this to get ethical board approval. And we got the ethical board approval around close to 2018, but at the same time, you needed to look at how to develop upscaling, and you have to make it to get a clinical approved product. And we started working in the GMP facility. This is, again, Dr. Larson, as you can see, the years are going on, and he came in 2019. This is in Nosral GMP facility. And by 2020, now around 2020, as Professor Wong said, I actually in London, I had to go to do my fellowship in pediatric urology. And we started getting, I got Dr. Anand, who's in the picture on the right-hand side, Dr. Yeo and Professor Wong, adult urologists, and for them to be a part of the project. And again, they were looking at other preclinical studies which were concurrently running while we were looking at the clinical trial. And finally, in 14th of August 2022, we had the implantation done in 2 patients. This is all of us outside the theaters, and we get Dr. Saiful from Hospital Serdang who came in to do the surgery with us. So this was about 8 years from the time I started my PhD and we started the initial animal experimental work. So I had to stress it's something that's, you need a team to work with. It's people from Switzerland, in Malaysia itself, between University of Malaya, we had the work done in UPM. We were kind to have the urologists from the Ministry of Health coming in to do it with us and also get us looking at patients for us, you know, and referring to our center. 8 years gone, well, the journey continues. And I don't know whether you remember the earlier picture. This was taken yesterday. That is Evie now. She's still working with us. We were sitting down in Gloria Jeans and got a piece of paper and a pen, talking to the clinicians here in UM about, you know, other projects, how do we go forward, other clinical indication. And again, that's with the research team working at, you know, looking at new projects and new things to do in UM. Thank you.
Thank you, Dr. Ganesh. Very, very happy for you, your achievement, your success. 8 years of a journey is not easy. You've just shown us snippets. I am sure you have, you know, all the difficult parts that you have just, you brush it aside and you continue and persevere and continue to work on. So I'm glad that also that AEU was able to assist you. Yeah, and now we have new projects. So thank you very much, Dr. Ganesh. I have a question from Professor Tunku Sara for you. Could you please summarize what is used to replace the urethral in a human now?
So Professor, at the moment, what we do is we take a oral cavity. So we take a harvest a tissue of the buccal mucosa, and then we use that. Or certain centers do pre-auricular. So even right now in London, when we have to do that, we do the surgery, we do the whole dissection at the level of the urethra. Then we go up, I have to flip the oral cavity. My anesthetist has got to move the tube the opposite side, and then I need to harvest a segment of tissue from the oral cavity to be used. So that's current gold standard. And it's actually very niche, few people do it. So most of these patients need to be sent to teams that can do this type of surgery.
All right, and Prof. Tunku Sara says, thanks for bringing in a lot of research into UM. Thank you, Dr. Ganesh. And I have another message here from Dr. Krishna. Good talk, Dr. Ganesh. I just want to know, what is your expectation in terms of duration for the smooth muscle cells migration to the implanted region? What could have delayed the migration of SMC at your early model?
So the thing was, that is the histology, that is immunohistochemistry. The animals were voiding. So functionally, the animals were voiding. We wanted to understand how we could improve based on our histology. So one of the many things that we found, and what Krishna's question was from that. So I kind of agree with what Prof. Fong said. I skipped a lot of stuff here. We went through a lot of different things, and the idea was how to improve the regeneration. So it's asking your question, what is the most important individual or cell type, and delivering that. It was the smooth muscle cells at the end. They were the king. They were the kingmaker. It was not urothelial. So what we had to do is fabricate something that the smooth muscle cells like. What smooth muscle cells like, the surgeons don't like. The material is not easy to work with at that time. But we realized that to get the smooth muscle cells in 3 months, in those cohort of animals, we got better outcome, meaning more of them were not having fistulas or strictures. So essentially, And that's what we did. We changed the material to suit the smooth muscle cells because the smooth muscle cell was the kingmaker. And we got them in coming in at around I did not share the current technology platform has shown in our preclinical animal model because I've not published it yet, but we're actually looking at even as early as 6 weeks where the cells already start coming in and having some layers of smooth muscle cells.
All right. I have a follow-up question from Professor Tunku Sara. I mean, what has changed after this research? She asked. What has changed after this research?
Well, at the current moment, we have not translated this to clinical practice yet. We have done 2 patients now. This is the first 2 we have done. We have another 3 more patients to continue. The team now has come back to University Malaya. Avi and Matthias they've actually gone on to spin this off, and they are looking at creating their technical files again in for submission to the FDA and also Swissmedic for more multi-center studies and things. But they want to do it with us, and that is something that I'm very, very proud of.
Okay, nice. I have no more question for you, except maybe one from myself. Dr. Ganesh, you started off with rabbit surgery. You did. I remember you did do some work with rodents. Can you tell us what made you decide that rabbit is it's so expensive, it's not easy to handle the animal, so can it be done in rodent?
So for the urethral work, it couldn't be done in the rodent model. We did experiments in the rodent model in AEU and also in mouse model for skin. We did the one offshoot study and also for the bladder. But for the urethra, even in literature, the smallest model is the rabbit model. So we were stuck with actually not having much of an option. But in AEU, we did do other experiments with regards to skin and bladder work using the rodent model.
And that worked, the bladder work?
Yes, so actually the material has worked in the bladder model. We have moved we did in the rodent model, we also did in the rabbit model in AEU, and the gold standard is actually using the pig model. Obviously, it's a bit more difficult to use the pig model here in Malaysia, so we did it in Belgium. It was implanted in 3 pigs, and it worked. So obviously, with trying to fund clinical research, getting grants are difficult. So the team has actually now focused on working on just the urethra, and then hopefully slowly coming up with concurrent work with the bladder.
All right, thank you so much. Exciting time coming ahead for you, right? I have a question here from Dr. Krishna. Could you please highlight the most critical challenges that you have faced during this translation process? Thank you.
Thanks, Krishna. The challenges was, when I started in 2014 as a clinician who has, to be honest, not really had much interest in research. I'll be very honest. Came in from Ministry of Health. I was not interested in research. I was sort of advised, would be the term I would say, to do a PhD, which gave me 4 years out of being a clinician. And that was the first challenge. The second challenge was the fact that I wanted to be acceleratedly stole in my PhD thesis defense. I remember telling, not in my defense, but actually, I want to propose on a bench to bid, and said, well, that's not going to happen in 4 years. You got to get your PhD first. Well, true and behold, it took me 8 years. But the challenges were trying to circumvent and, working with a lot of different people from a lot of different backgrounds. And that's actually was my success. I did not do all of this alone. It was the guys there, the AU team, even people at Nosral, the urologists, my colleagues, friends. It was a very team-based effort that actually got it across. So those were my challenges overcoming that to get this to where it is today.
All right. Thank you, Dr. Ganesh. All right, so we have no more question, not even for EM, no question for EM. Okay, so I think thank you very much to all our speakers today, Dr. Prof. Louis, Dr. Prof. Lim, and also Dr. Ganesh. Thank you all for spending your time here and talking to young researchers, young clinicians here on the work that you have done. All right, so we'll end the session. Anything from you, Dr. Anwar?
Nope, just thank you everyone for joining us today.
Thank you so much. See you around.